I want you to imagine, just for a second, that you are standing in a cutting-edge laboratory.
Okay.
You lean over a microscope and you look down into this tiny clear dish. And floating in the nutrient liquid, there is a speck of tissue, maybe, I don't know, the size of a lentil.
Right.
But this isn't just any tissue. It's a self-organizing, three-dimensional model of a developing human brain.
Yeah, I mean, it sounds incredibly futuristic, right? But neural organoids are very much a reality today.
They really are.
They are fundamentally changing how we study early human brain development and, of course, devastating neurological conditions.
Absolutely. But, diving into the research for this deep dive, there has been this massive, almost unspoken problem hovering over the entire field.
Yeah, the validation issue.
Right, exactly. It reminds me of engineering. Like, if you were building a suspension bridge, you know the exact tensile strength of your steel. You can test your concrete, right? The materials are completely quantifiable.
Right. The properties are established long before you ever build. But when you step into the world of cultivating human brain tissue in an artificial environment, scientists have essentially been flying blind.
Yeah.
The central question has really always been, how do we actually know that the cells we coax into growing in a petri dish are truly a one-to-one match for the primary cells of a real human brain?
Which brings us to the core mission of our deep dive today. We are exploring a monumental achievement from an international team of researchers who decided to, finally turn the lights on.
They really did.
They've built an integrated transcriptomic cell atlas. Essentially, they created a master map that aligns what we are growing in the lab with actual biological human brain development.
And this atlas, I mean, it represents a total paradigm shift. Up until now, assessing the true biological fidelity of these lab-grown models has been, it's been like trying to put together a massive jigsaw puzzle without the picture on the box.
Oh man, that's a perfect way to put it.
We had all the pieces, but no reference for the final image.
So by the end of our conversation today, you are going to understand how scientists are finally separating the true biological signal from the artificial noise in these lab-grown organs.
Yes.
And crucially, we'll see how this master map is acting as a massive accelerant for discovering treatments for complex neural diseases.
It's super exciting stuff.
It is. But let's start with the root of the issue. How do we even grow a brain in a dish in the first place?
Right. So broadly speaking, researchers use pluripotent stem cells. And there are two main philosophical approaches to guiding them, which are unguided protocols and guided protocols.
Okay, so unguided sounds a bit like, I don't know, throwing seeds in a garden and just letting nature take its course, just providing the soil and water.
That is actually a highly effective way to think about it. Unguided protocols rely entirely on the innate self-patterning capacity of the stem cell.
So they just know what to do.
Basically, yeah. You provide a supportive liquid medium, and the cells naturally start to divide and differentiate into this wild diversity of cell types, much like they would during early embryonic development. They self-organize without us telling them what to become.
But I'm guessing that approach doesn't give you a lot of control if you want to study, say, a very specific part of the brain, which is where the guided protocols come in.
Precisely. With guided protocols, researchers take a highly active role. They introduce external patterning factors into the nutrient bath.
Okay.
These are specific chemicals called morphogens. Think of morphogens as molecular street signs.
Molecular street signs, I like that.
Yeah, by introducing certain morphogens at very specific times, you can actively steer the development of the organoid toward becoming a specific brain region, like the cerebral cortex or the midbrain.
But here's where the fragmentation problem really begins, right? Because you have hundreds of research institutions globally.
Oh, absolutely.
Some are using unguided methods, some are using guided methods, and everyone seems to have their own, unique cocktail of morphogens and nutrient broths.
Exactly. Every single lab was creating its own isolated ecosystem. And when they published their findings, the data was structurally completely different from the lab down the street.
It reminds me of, you remember the boom in sourdough baking?
Oh, yeah, of course.
Imagine thousands of bakers all over the world all trying to recreate an ancient sourdough recipe. Some use rye flour, some use wheat, some let it rise in a warm room for a day, some put it in the fridge for a week.
Right, they all end up with bread.
They are all technically making bread, but without a master database to compare every single recipe and the final chemical makeup of the loaves, you have no idea whose bread is actually closest to the original recipe.
That's spot on.
Until this project, nobody had that master database for brain organoids. Okay, let's unpack this. How do you even begin to compare nearly 2 million cells from dozens of different labs?
Well, to build that database, which is called the Human Neural Organoid Cell Atlas, or HNOCA, the researchers had to operate at a scale that is just difficult to overstate. They gathered 36 different single-cell RNA sequencing datasets.
Let's clarify what that single-cell RNA sequencing actually means for the listener.
Yeah.
Because it's not just taking a microscope picture. It's literally cracking open individual cells to read their active messenger RNA. Right.
Yes, exactly.
It tells you exactly which genes are turned on or off at that exact moment.
Yeah, it provides a high-resolution barcode of a cell's current biological state. And they collected this data across 26 distinct growing protocols.
Wow.
Yeah, incorporating organoids that were grown for as little as 7 days all the way up to 450 days.
And when you combine all of that, you get a staggering 1.77 million individual cells.
It's massive.
But having nearly 2 million cells from dozens of different labs introduces a massive logistical nightmare. If Lab A sequences their cells on one type of machine and Lab B uses a different machine in a totally different climate, won't the RNA readouts show differences that have nothing to do with biology, but everything to do with the room they were in?
You've identified the absolute bane of computational biology. We call those batch effects.
Batch effects.
Right. A batch effect is any technical difference in your data caused by how, when, or where the experiment was run. If you don't remove those batch effects, your algorithm might group all the cells from Lab A together and all the cells from Lab B together.
And completely miss the underlying biological similarities.
Exactly.
So how do you iron that out? Because the methodology here is the magic that makes this entire master map possible. The paper outlines a very specific 3-step computational pipeline.
Yes. The first step relies on something called a reference similarity spectrum, or RSS. They took this massive chaotic pile of organoid data and mathematically projected it onto a pre-existing, highly validated atlas of the actual primary developing human brain.
It's almost like taking a piece of semi-transparent tracing paper with all your messy lab data scribbled on it and laying it over a highly detailed, perfect satellite map of a real human brain just to see what roughly lines up where.
That spatial alignment is the perfect starting point. Once they map those rough coordinates, they move to step 2, deploying an algorithm they built called Snapseed.
Snapseed.
Yeah, and this handles the preliminary marker-based annotation.
So marker-based goes back to those RNA barcodes we talked about earlier. It's looking for specific genes that are always reliably turned on in specific types of cells.
Right. So if a cell is expressing a specific protein signature that is unique to, say, astrocytes, Snapseed recognizes that barcode and tags it as an astrocyte.
Okay, that makes sense.
It establishes the basic cellular landscape. But that brings us to the crucial third step, which handles those pesky batch effects we just talked about, using a tool called SciPoly.
SciPoly, this is the label-aware data integration step. This is where we scrub the technical noise. But how does an algorithm actually know the difference between a real biological mutation and a technical error caused by a lab technician using a different brand of test tube?
It's a great question. SciPoly achieves this by mapping the cells into what we call a latent space.
Space.
Right. Instead of just looking at the raw RNA data, the algorithm creates a multidimensional mathematical space. It looks at the Snapseed tags from the previous step and learns to prioritize the core biological function over the superficial lab dialect.
Oh, wow.
Yeah, it figures out that an astrocyte from Lab A and an astrocyte from Lab B, despite having slight technical variances in their data, share the same fundamental vector in this latent space. It mathematically pulls them together into a unified, standardized format.
So it basically translates dozens of different lab dialects into one universal biological language.
Exactly.
And the final output of that massive computational pipeline is the HNOCA. We finally have our master map. Let's do a quick recap for you, the listener, because we just covered a ton of ground.
Good idea.
We've got unguided and guided protocols for growing brain cells, which created a huge, disorganized mess of data.
Right.
But by using a 3-step pipeline, the tracing paper RSS, the Snapseed barcodes, and the SciPoly latent space translator, they've cleaned the data and built a 1.77 million cell master map.
That's the perfect summary.
And with that unified map, the researchers could finally ask the critical question: how accurate are our artificial brains compared to the real thing?
And the findings there revealed a massive geographic discrepancy when they measured the transcriptomic similarity.
Meaning how closely the RNA of the lab cells match the primary human cells.
Yes, exactly. They found a stark spatial bias. Cells derived from the dorsal parts of the brain were highly accurate, but cells derived from the ventral parts were not.
To visualize this, dorsal regions are generally the top and back parts of the brain structures, right?
Hmm. Yes, the dorsal telencephalon, for example, is where our excitatory neurons originate. Ventral regions are the bottom or front, giving rise to different structures, including many of our inhibitory neurons.
And the data shows that across the board, regardless of the protocol, we are really good at growing the top of the brain in a dish, but we are struggling to accurately mimic the bottom.
Unfortunately, yes. The guided protocols designed to mimic the ventral regions showed a consistently lower transcriptomic similarity to real human development.
I have to challenge the utility of this a bit.
Okay, go for it.
Wait, so if you are a scientist dedicating your career to studying a neurological disease that originates in the ventral part of the brain, say, certain developmental pathways involved in schizophrenia, are these organoids basically useless to you right now? Doesn't this just prove that your models are fundamentally flawed?
This raises an important question, and it is tempting to look at that spatial bias as a failure of the field. But identifying this limitation is actually what makes the atlas so incredibly valuable. Yeah, previously researchers didn't definitively know why their ventral models were behaving strangely, or even exactly where the biological divergence was happening. They were just guessing.
Ah, I see. It's the difference between knowing your car engine is running rough and plugging it into a diagnostic computer that tells you the exact oxygen sensor in the exhaust is misfiring.
Exactly. You can't fix the engine until you identify the specific point of failure.
And now they have that precise diagnostic target.
Yes. By pinpointing exactly where the current guided morphogen protocols are falling short, scientists know they need to refine their chemical recipes. They can experiment with new morphogen combinations and use this very atlas to test if their new recipes are pushing the ventral cells closer to true human biology.
So the map isn't just a validation tool, it's a diagnostic roadmap for future engineering.
Precisely.
And moving beyond just the geographic differences, the atlas uncovered another layer of noise. They found a universal molecular anomaly hiding in almost all of the lab-grown neurons, regardless of whether they were dorsal or ventral.
Right. They identified a persistent signature of cell stress across the in vitro neurons. Specifically, they showed a perturbed metabolic signature heavily associated with glycolysis.
Glycolysis, that's the metabolic pathway that converts glucose into energy.
Right.
It's fast, but it is not the standard, highly efficient way a mature brain cell normally gets its power.
No, not at all. In a developing primary human brain, neurons quickly transition to a more complex energy process called oxidative phosphorylation.
Okay.
And that requires a robust and steady supply of oxygen delivered through a network of blood vessels. But lab-grown organoids don't have a vascular system. They don't have blood pumping through them. They are simply floating in a nutrient bath, relying on diffusion.
Ah, so because they lack that vascular oxygen delivery, they're forced to rely heavily on glycolysis just to survive.
Exactly.
It reminds me of a stressed-out college student pulling all-nighters during finals week.
That's a great analogy.
Right. They are removed from their natural, healthy environment, and instead of eating a balanced diet, they are surviving entirely on a sugar rush of energy drinks just to keep the lights on.
The organoid cells are essentially trapped in a metabolic sugar rush due to the artificial constraints of the petri dish. And for a long time, this stress signature was severely muddying the waters.
Because if you sequence that stressed-out cell, the RNA barcode looks completely different from a calm, healthy cell in a real human brain.
Yeah, it made it look like the core genetics of the lab-grown neurons were just fundamentally wrong.
But because the researchers had this massive integrated data set, they could apply advanced mathematical modeling to filter out that specific noise. They treated the metabolic state and the maturation state of the cells as covariates.
What's fascinating here is how they handled those covariates.
So covariates being variables that you can isolate and mathematically subtract from the equation.
Right.
You essentially write an algorithm that says, show me what this cell would look like if it wasn't currently experiencing a finals week sugar rush.
And when they factored out that environmental stress, the results were astonishing. The number of differentially expressed genes, the genes that appeared to be behaving incorrectly in the lab versus the real brain, plummeted.
Oh, wow. So underneath all that superficial and environmental stress, the actual machinery of the cells was perfectly intact.
Exactly. When you look past the glycolysis, the fundamental biological functions are highly faithful to primary brain development.
That's incredible.
The paper specifically highlights the synaptic vesicle cycle, which is the precise mechanism neurons use to package and transmit chemical messages across synapses.
Right.
They also looked at the negative regulation of calcium channels, which dictates how neurons fire electrical signals. That core communication machinery is a remarkably accurate one-to-one match.
There has to be a massive sigh of relief for the entire field of neuroscience. It proves that despite the artificial non-vascular environment of the dish, the foundational biology of human cognition is actually forming correctly.
It completely validates the use of these models for studying deep biological functions.
Okay, so let's do another quick recap before we hit the final stretch.
Sure.
We learned the map showed us dorsal cells are great, ventral cells need work.
Right.
And we learned that the lab cells are all super stressed out, running on glycolysis.
Yeah.
But once you mathematically erase that stress, the core neural machinery is spot on.
That's exactly right.
So let's pivot to the clinical reality, because this is where the Atlas transitions from being a brilliant piece of computational biology into a tool that could actually save lives.
Yes.
We have the map, we understand the dorsal ventral limits, and we know how to mathematically erase the metabolic stress. How is this being applied to human disease right now?
Well, the researchers took this massive integrated atlas and transformed it into a universal control cohort.
Okay.
And to understand why that is so revolutionary, we have to look at how disease modeling traditionally works.
Right. So usually if a lab wants to study the mechanisms of Alzheimer's, they take stem cells from a patient with Alzheimer's, and they spend months growing a diseased organoid.
Yes.
But to actually spot the anomaly, you have to compare it to a healthy baseline.
Which means that same lab has to spend equal time and resources cultivating a perfectly matched, healthy organoid to act as their control.
Which sounds exhausting.
It is incredibly time-consuming, highly expensive, and completely vulnerable to those batch effects we discussed earlier.
Oh, right.
Your diseased organoid might look different from your healthy control just because the incubator temperature fluctuated, not because of the Alzheimer's.
Here's where it gets really interesting. With the HNOCA, they don't have to grow the control from scratch anymore. The researchers collected 11 distinct single-cell data sets that modeled 10 different highly complex neural diseases.
Yeah, we're talking about pathologies like autism, schizophrenia, Alzheimer's, and glioblastoma.
So instead of relying on their own isolated healthy controls, they took the data from those diseased models and projected it directly onto the HNOCA atlas.
Exactly. They overlaid their disease tracing paper onto the aggregated average of millions of healthy cells from dozens of independent labs.
And by doing so, they could instantly identify the specific genes and developmental pathways that were deviating from the massive, standardized, healthy baseline.
Right. They bypassed months of control cultivation and almost entirely eliminated the risk of batch effects clouding their judgment.
Wow.
They could see the exact molecular machinery causing the pathology.
For something like glioblastoma, a highly aggressive brain cancer, being able to project a tumor model onto a perfect map of healthy development to see exactly which genetic switch flipped. I mean, the efficiency of that is incredible.
It's game-changing.
It accelerates the timeline from basic observation to potential drug screening by an order of magnitude.
It accelerates discovery globally. And if we connect this to the bigger picture to ensure that acceleration reaches everyone, the research team didn't just publish their findings in a static PDF and move on to the next project. Right. They built HNOCA tools.
HNOCA tools is a programmatic interface, right? It's software.
It is an open-source analytical suite. It allows any researcher anywhere in the world to plug their own newly sequenced RNA data into the exact same computational pipeline we just discussed.
So if a lab in Tokyo develops a brand new morphogen protocol to try and fix the ventral brain issue, they don't have to wait years for someone else to validate it.
No.
They sequence their cells or run it through HNOCA tools and instantly see where their new recipe lands on the master map.
And as they project that new data, the atlas itself can evolve. As the global community feeds it new protocols, new disease models, and organoids grown for longer time periods, our understanding of both healthy and perturbed biological states continuously updates.
So it's not just a map, it's a living, breathing resource that grows denser and more accurate every single day.
Exactly.
Which brings us all the way back to where we started. That tiny, lentil-sized speck of tissue floating in a dish. We started by looking at it as a mystery. A dark room where we weren't quite sure if the cells we were growing had any real relation to the human mind.
But by leveraging 1.77 million individual cells and some brilliant mathematics, We have turned the lights on.
We really have.
We've mapped the geography, corrected for the artificial environment, and proven that the core synaptic machinery is a stunningly accurate reflection of us.
It really is a triumph of separating the signal from the noise. And it leaves me with this final thought, something for you to mull over long after this deep dive ends.
Let's hear it.
If we have truly reached the point where we can perfectly map a developing human brain, mathematically erase the stress of the petri dish, and simulate the exact genetic pathways of complex diseases to a one-to-one transcriptomic match.
Yeah.
At what point does our computational and cellular model stop being just a model and start becoming an actual functional synthesis of human biology? Is a fascinating line to walk.
It certainly challenges our traditional definitions of biology.
It really does. Thanks for joining us on this deep dive into the Human Neural Organoid Cell Atlas. We'll catch you on the next one.